Dr. Lucy Sullivan, who is currently the Scientific Director, Transplantation and Immunogenetic Services, Australian Red Cross Lifeblood, and Lab Manager in South Australia for the Transplant and Immunogenetic Service. Lucy actually used to work here in Melbourne, was a researcher with our team specifically, but also at Peter Doherty, but has now transitioned over to South Australia. We welcome Lucy back to Melbourne.
Thank you, Bron. So, hello everyone. Welcome today. I hope I'm not going to overwhelm you with too much immunology. I'll try to go easy on you, but thank you for the opportunity to talk here today. So I've already been introduced, so I'm not going to go into who I am, but just to let you know, I'm from the Lifeblood Transplantation and Immunogenetic Services, and because that's very long, we call ourselves TIS. And I'm going to be talking to you today about improving donor-recipient matching in lung transplantation. So I thought I'd just start off by telling you what is the TIS services. So we're a highly specialised pathology service, if you like, where it's recipient-focused diagnostic testing for both solid organ and stem cell or cord blood transplantation. And we also do specialised genetic testing for disease associations, for example. And so our scientists are highly trained and highly specialised. It takes a long time for TIS scientists to really get to senior positions, and they provide both laboratory results and clinical advice for the compatibility assessments between recipients and potential donors. And so there are 5 transplantation testing laboratories in Australia, and the 3 largest are in Lifeblood, being in Sydney, Melbourne and in Adelaide, where I am. And the 3 Lifeblood services cover 80% of all transplantation in Australia. So there are also another tissue typing service in Queensland and WA, and they sit under their relative pathology services rather than in Lifeblood. So what do we do? What are these diagnostics we do? So whenever there is a recipient and we have potential donors, we need to do extensive testing and data analytics to determine the degree of compatibility to prevent rejection. So when a patient gets listed for transplant, they go on a national and state waiting list, and these waiting lists are managed by the TIS labs. And transplant compatibility is determined through HLA compatibility of the recipient and donor. So I'll just go into a little bit about what is HLA. So HLA is human leukocyte antigens. So these are proteins that are expressed on the surface of most of your cells in your body. And what they are designed to do is to tell the immune system when you've got a foreign body coming in, so such as like viruses or infection, and to prevent any sort of negative impacts from viruses, to fight them off and to get rid of them from the body. So it sort of alerts the immune system that something's a bit foreign. But in the case of transplantation, the donor HLA is actually foreign to the immune system. So this is called HLA mismatching. So HLA mismatching between donors and recipients promotes an immune response and transplant rejection. So I guess you'd ask, well, why don't we just match donors and recipients? Well, the problem is that HLA is extremely different between different people. So as I put here, so over 99% of our genes are the same, but your HLA type accounts for much of the difference. So given there are a huge number of different HLA, it's very difficult to find a complete match between donors and recipients, unless it's somebody very closely related to you. So what do we do to determine compatibility rather than, I guess, matching between donors and recipients? So as I said, HLA testing is performed only in our labs. It's not in any other pathology services in the country. So we look at HLA typing for both donor and recipient by DNA-based assays. And we also look for the presence of HLA antibodies in recipients. So antibodies to a donor are bad for transplant and can result in rejection, so we want to avoid them. So these HLA antibodies can develop by pregnancy, transfusion or previous transplant, and we monitor them for both pre and post-transplant in the recipient. So as mentioned, so antibodies to a particular donor are called donor-specific antibodies. And we also perform cross-matching between donors and recipients. So this is the most complex type of testing that we do. And this is where we look at individual compatibility between a particular recipient and a particular donor by mixing the patient's serum that contains or may not contain the HLA antibodies with the donor cells to see if the antibodies present in the patient's serum are capable of interacting with donor cells, and if they are, that could be bad for transplant. So the way that we do that these days is by a flow cytometry cross-match. And I've highlighted these two terms here because I will be talking about them later, so I sort of want you to remember them. And a positive cross-match reflects donor-recipient incompatibility, so we want to avoid that. And in our labs, there's minimal automation, so the testing is really specific for each patient, and it's also ongoing. We need to monitor regularly for the presence of HLA antibodies in our recipients. So we've really been supported across all of those testing areas by the Lungitude Foundation. I'm very grateful for their support, and in particular for this funny-looking fellow here, who I hope's online, called Steve Hi-Ho. So he did a PhD under my supervision and with the Alfred team, and he had an amazingly successful PhD. He's actually still going, but not to scare you with the titles of the papers, but just to let you know. So he has published a number of papers in his PhD. This is a huge number of papers, he's not even finished yet. And I'm just going to focus on this last one here, because it really did change the way we did things across all of the labs in Australia. So I guess to do that in a PhD is pretty amazing. It was all with the support of Lungitude that this happened. So I just need to go in a little bit first about the virtual cross-match. So it sounds all very, sci-fi. It's not really. What it means is we're not actually using donor cells to look at a particular donor-recipient compatibility, but we're looking at the antibodies that are present in a recipient and then looking at the donor HLA type. So we can do this to predict if a cross-match will be, if we can predict whether we will have a positive or negative cross-match based on the level of antibodies in the recipient. So how does this work? If we have, just for an example, don't worry about the numbers, but a donor who has a HLA type of A1B7, and we have a couple of potential recipients here, and they both got antibodies. So recipient 1 has antibodies to A1 and B7, and recipient 2 does have antibodies, but they're not to the donor. So we can say that this particular recipient is more compatible than this recipient because this recipient has donor-specific antibodies and this one doesn't. So that's really what a virtual cross-match is, where we compare the donor HLA type and recipient anti-HLA antibodies. So if they're antibody negative, they're compatible. If they're antibody positive, they're incompatible. But we really wanted to test this over here because sometimes we are limited in the availability of donors, or there may be a particular urgency. So we asked the question, are all DSAs compatible? Or more importantly, can the strength of an antibody actually be used to predict the flow cross-match, which is also called FXM, result? So this is what Steve did. So he did a huge number of flow cytometry cross-matches using different recipient sera, and these were purposely selected based on the presence of different HLA antibodies. And then he selected donor cells from transplant, donor cells from different HLA types, and these weren't donors that necessarily went to transplant, but just a different number of HLA types, so we could test the different recipient sera to see if they were cross-match positive or cross-match negative. So you performed the flow cytometry and then looked at the association between the antibody strength and the flow cross-match reactivity. And we do two different types of cross-matches on T cells and B cells. And then he wanted to determine the threshold for when it was, I guess, compatible or not compatible if there were lower level antibodies that we could potentially cross with a donor. So these are results here. So we've got the T cell and the B cell cross matches and whether antibody negative or antibody positive. So Steve defined a threshold based on the antibody strength of all of these flow cross matches that he performed. So I just wanted you to concentrate on the ones in red, because these are the ones where they're either both negative or both positive. So you can see that by far there is a very large concordance between when they are both negative or when they are both positive compared to the other quadrants here. So in actual fact, we found that about 90% of the time we could actually predict whether a cross-match was going to be positive based on this threshold that Steve defined. So this was really good news because it created a bit of a nuance here. So now instead of them all being incompatible, if you like, if they had antibodies that were less than this threshold, they were considered compatible, and greater than the threshold, they were considered incompatible and would not proceed to transplant unless it was really an emergency. So the conclusion and translation of these results was that we define an antibody threshold to accurately predict a cell-based cross-matching technique. So why would we do this? Why would we do virtual cross-match? Well, it does provide a much faster assessment, so we can do it on the night of transplant really quickly and easily, as opposed to a flow cross-match that takes a lot longer. And it also doesn't rely on donor cell quality. So sometimes from the donor, we can't get blood cells to actually do a cross-match. But with this threshold, we can now still predict whether it's going to be positive or negative in the absence of the donor cells. We can also assess more recipients per donor to determine sort of a hierarchy, if you like, of compatibility. And it also provides improved access to transplant for patients who have lots of antibodies. So some of them we know we can now cross and some of them we can't. And virtual cross-match is now used nationally for the assessment of transplant risk in both not only lungs, but heart, liver, kidney, and pancreas transplants. And really importantly to note is that this particular research that I presented today was critical in the rollout of virtual cross-match nationwide, so not just in the TIS labs, but Australia-wide. So I guess it's just a testament to the support of Lungitude, and it's really made a change in clinical practice for us. So what are we going to be doing in the future in the Lifeblood labs? We want to look at some novel ways of monitoring rejection post-transplant, and Glenn's briefly touched on this, but one thing that's gaining momentum, particularly in kidney transplantation, is looking at donor-derived cell-free DNA in the blood of recipients. So I guess very simply put, the theory is that if the organ is suffering from rejection and getting damaged, they get more donor DNA into the blood of the recipient, and you can measure that. And it can give you an indication that something's going a bit wrong and gives you an opportunity to intervene earlier. The other one that we would like to look at moving forward is the assessment of antibodies to non-HLA. So I've spent a lot of time talking about antibodies to HLA. It's also a whole suite of antibodies that conform to non-HLA targets. And we know from kidney transplantation that these antibodies, these non-HLA antibodies, are also associated with rejection and decreased graft survival. And in particular, there's been a lot of research around antibodies to this molecule called AT1R. However, it's unknown what their clinical impact is following lung transplantation. So just very briefly, I'm going to sort of talk to you about AT1R testing. My lab in SA is the only lab in the country that looks at for antibodies to AT1R. So currently we look at AT1R antibodies pre-transplant for only SA, kidney and kidney pancreas patients. And we just started looking at pre-transplant AT1R antibodies in lung transplant waitlisted patients, and at this stage it's Alfred only. We also do quite a bit of post-transplant testing, normally when clinically indicated. So this is when transplant recipients are showing signs of rejection, but we can't find any HLA antibodies. We then often look for AT1R antibodies, and they often come up positive. And we also do look at a lot of research samples. So for kidney transplant recipients, this is current protocol in South Australia, based on some research that was done a little while ago. And it's been published, I think, back in 2016. So what we found was that high levels of this AT1R antibodies found pre-transplant were significantly associated with the development of rejection in kidney transplant recipients. But they're only present in a relatively small number of patients. So based on this research, the local kidney transplant team changed their protocol to preemptively treat patients with high levels of AT1R antibodies prior to transplant. And coincidentally, since that time, the rejection rate has fallen from 30% to 8%. So At rejection, AT1R levels are much lower than pre-transplant, so therefore we know we have to look at pre-transplant sera for the assessment of risk associated with AT1R antibody levels. So we want to now start looking to see if this is also translated into our lung transplant cohort. So we are going to embark on a study to determine if non-HLA antibodies contribute to outcomes following lung transplantation. And particularly we want to look at clinical parameters such as rejection, kidney function, as well as lung function and ICU stays. So this could offer an opportunity for therapeutic intervention, much like in the kidney transplant cohort in SA, and hopefully also further improving survival rates following lung transplantation. So that's it from me. Once again, thank you to the Lungitude Foundation for the ongoing support. And the research I presented today was really driven by Steve Hyho, who is almost finished his PhD, we hope. He's been spectacularly successful, so it's been an amazing student to have. Also from our own lab, Rob Carroll, Sue Dayton and Tim Emery have really been driving the AT1R research. And very close collaboration, even though I no longer actually work with them, all my colleagues at the Alfred Hospital. And also a big thank you to the lung transplant patients who generously donate blood samples and BAL samples for our research. So without their consent, that wouldn't be possible. So thank you.